Hello, everyone, and welcome to today's webinar, Expanding Proteomic Horizons with Single Molecule Proteome Analysis. I am Cassie Soltman of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Nautilus Biotechnology. To learn more, visit nautilus.bio. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. Also, if you experience any technical issues, you can utilize this Q&A box and we will assist you directly. I'd like to now welcome our speaker, Dr. Parag Mallick, founder and chief scientist at Nautilus Biotechnology. Parag, you may now begin your presentation.
Thank you so much for the introduction. I'm really excited to be here today to share with you some of the latest exciting data from Nautilus. Before we dive in, I'd like to start off by just recognizing how tremendous advances in biology have arisen from advances in resolution and scale. We saw this in protein structure initially in the transition from looking at diffraction maps ultimately to low resolution crystal structures, to where we are today, where we have high throughput methods to solve crystal structures at sub-angstrom resolution. And that enabled our understanding of biology and protein interactions and drug protein interactions. We saw this again in omics, is the transition from looking at bulk populations of either proteins or transcripts or DNA to a transition to looking at single cell, and then even more recently with a transition to looking at spatial. This also happened in proteomics itself with the evolution from looking at 2D gels to mass spectrometry. And what I'm excited to share with you today is another jump in resolution and scale. What we're going to cover today are a couple things. We're going to start off with talking about the platform that we're building, why we're building it, and introduce you to the fundamental aspects of it. We'll then talk about how the platform can be used in a broad scale manner to look broadly at the proteome. And then we'll dive into the other aspect of the platform, which is really looking in extremely high resolution at particular target protein molecules, in this case, tau. Coming back to the beginning, Nautilus was formed with the idea that we wanted to develop an entirely novel platform for proteomic analysis that was complementary to existing tools, but rather than being an optimization, really was a ground up design. And when we were doing that thinking, we started and we said, all right, well, what's our criteria? What do we want our new proteomics platform to potentially be able to do? And we came up with this shopping list. I'm really curious to hear from each of you about what your desires would be for a next generation proteomics platform. But for us, what we really wanted were, number one, something that was comprehensive, able to substantively measure all of the proteins and proteoforms in a sample. We wanted something that it was exquisitely sensitive to be able to look at those incredibly rare and important protein molecules, such as transcription factors. We wanted something that had a wide dynamic range that matched the scale of the proteome. As an analytical tool, it needed to be reproducible and robust. And then ultimately, wanting to really explore biology and crunch through large numbers of samples, we wanted to have a rapid runtime. Lastly, a really critical aspect of our platform that we wanted to build was something that was easy to use, something that was accessible to the broader biological community, not just to the extant mass spectrometry community. As we thought about ways to accomplish some of these goals, we arrived at building a single molecule-based platform. And there were a couple of reasons for that. The first is, definitionally, one cannot be more sensitive than a single molecule platform, with no translation to intensity or brightness or any other lossy transform. Additionally, what's really special at the single molecule level is that it changes the nature of measurement. In particular, if I can identify each individual molecule, I get my quantification for free. I just count up those identifications. So the nature of the challenge at the single molecule level is one of identifying each individual molecule. One other special aspect that we wanted in the Nautilus platform was one that operated on undigested proteins. What this enables is, unlike with peptide-centric methods, where in order to do quantitation, one has to do peptide to protein inference and reassembly, which can make quantification challenging. Another key aspect is that when we want to be able to look at proteoforms, the patterns of multiple modifications on individual molecules, that information is totally lost when looking at digested peptide fragments. And so there is no way to really get to proteoform information without looking at full-length undigested proteins. Now, the platform does denature the proteins, but it does not digest them. In building such a platform, there are several specific challenges one has to overcome. The first is you have to guarantee that you're looking at single molecules. Second, one has to identify each of those molecules. And then third, one has to perform all of these measurements fast enough to have both incredibly high throughput and sufficient dynamic range. So we'll start by walking through how we've been addressing these challenges. This first question of how do you guarantee single molecules? One of the most common techniques that people use for getting to single molecules is a technique called limiting dilution. In this technique, samples are diluted and then they're spread out over a giant surface. And on that surface, one of the challenges is that when you do this, most of the space is empty. And so if you're scanning across this surface, you're spending most of your time measuring empty space. That's not great for efficiency and throughput and measuring a large number of molecules. The other challenge with Poisson-based loading is that you actually can't guarantee that you're looking at individual molecules. In fact, 50% of the locations where there's any molecule, there'll be multiple. 25% will have 2 molecules, 12% will have 3, and on and on and on. And so this was the first challenge we had to overcome, which was how do we actually generate a hyperdense single molecule protein array? The ways that people typically approach this problem are by creating landing pads for individual protein molecules that are really small so that you don't have multiple molecules living at the same coordinate. Our team came up with a really clever approach, which goes the other direction. Instead of trying to make landing pads that are small, we instead make the proteins big. And we do that through the use of an intermediate nanoparticle, which is shown here. This nanoparticle is really special. It's something that was designed inside our building, and it looks exactly like it's shown here. It's sort of a brick with a flagpole sticking out the top. And at the top of the flagpole is a click chemistry reagent known as a tricyclooctene. There are no other chemically labile functional groups on this entire molecule. This is really different from a standard nanoparticle of this size, about 100 nanometers. A standard nanoparticle would have hundreds of thousands of conjugation sites, and so it could hold hundreds of thousands of protein molecules. Instead, these nanoparticles that we've designed can hold exactly 1 at the top of the flagpole. So the library preparation, sample preparation method consists of first taking our sample proteins, functionalizing the primary amines alongside denaturing, so all those lysines and N-termini, using an STP ester that has a methyltetrazine group on it, a click chemistry reagent group. We then mix those functionalized proteins, those labeled proteins, with the nanoparticle scaffolds. The functionalized proteins then conjugate to those nanoparticles. Again, the nanoparticles can only hold exactly 1 molecule. We then take those nanoparticle protein conjugates, flow them over a specially nanopatterned flow cell, and that nanopatterned flow cell has these little landing pads, or large landing pads, that are sized to match the size of the nanoparticle. And each one can only hold 1 nanoparticle. So what you then get is you get 1 nanoparticle per landing pad, and 1 molecule per nanoparticle. one protein molecule per nanoparticle, which then self-assembles into a hyperdense single molecule protein array, essentially taking every protein molecule from the sample and depositing it on its own special coordinate on a hyperdense single molecule array. This is upside down of how many traditional approaches work, where they will instead have bulk affinity reagents immobilized on a surface. We are taking every molecule from the sample and immobilizing it on the surface. For a little more detail about what the actual sample preparation, library preparation workflow looks like, shown here, you can see that this is done essentially as a one-pot reaction, where we start with the sample, somewhere between 150 nanograms and 10 micrograms of protein. We then we functionalize the proteins, denaturing along the way, and then we also modify and block the lysines. We then do some cleanup steps, mix in the scaffolds, and then do another cleanup to remove free protein, and then flow it over the flow cell to generate this single molecule protein array. Shown here is a zoom in of what one of our flow cells look like. And each one, what you can see here is we've zoomed in one level of detail, we've further zoomed in, and each one of these little illuminated blobbies is one protein molecule. This is so important because this allows us to both guarantee that we're looking at single molecules and also arrange them in a hyperdense manner that allows for efficient measurement. Just for sense of scale, each one of these squares, each one subarray, contains about 125,000 molecules. The flow cell overall has been designed so that it has about 1 billion molecules per lane, so measuring about 10 billion molecules overall per experiment. We'll talk about that more later, but that scale is really critical to enabling a wide dynamic range measurement. One of the questions that we also wanted to verify was that our library preparation process did not substantially bias what proteins in the sample ended up depositing on the flow cell. That we didn't, for instance, select away from proteins that were membrane proteins or were small proteins or large proteins. To examine that, we did a number of different experiments, but I'm showing one here where we started with a control mixture of a mix of 48 proteins, known as the Universal Protein Standard 1. We analyzed that mixture on its own. We then put it through our library preparation process and then released the proteins that had been conjugated to the nanoparticles, and then analyzed the proteins afterwards and showed that there was no significant difference in the protein composition before and after our library preparation process. We also did similar experiments with whole cell lysate as well and showed that the biases introduced by our library preparation method are modest and not greater than introduced by current proteomics sample preparation workflows. As I mentioned, one of the most important aspects of our platform is its scale. From day one, we knew that we would want to measure billions of individual protein molecules. And as I alluded to, the number of protein molecules is directly related to the dynamic range of the platform. To examine this, we did simulations to examine how, depending upon the number of molecules analyzed, what that led to in terms of sampling of molecules from a sample deposited on the flow cell. And what you can see here on the right is that relationship. In orange is shown the dynamic range as measured between the least abundant protein in a cell sample and the most. And you can see that there's a sweet spot around 10 to the 9 or 10 to the 10 whole proteins measured, undigested proteins measured. I'll note that if we were digesting the proteins, each protein gives rise to around 100 or so peptides. And so even if you were measuring this many molecules, if you're doing it at the peptide level, your dynamic range would be 2 orders of magnitude less. Once we've immobilized the sample onto the flow cell, the next question is, how do we measure it? And how do we identify what each molecule is? Zooming into the broader workflow under the hood of the Nautilus platform, it really consists of 3 pieces. The first is what we've just talked about, this single molecule library preparation and deposition step. The second is about iteratively probing each molecule over and over and over again using fluorescently labeled affinity reagents. And then the third aspect is the data analysis and interpretation. So if one zooms into the instrument, what occurs at each step is that we first immobilize the sample proteins in the flow cell. We next introduce a series of fluorescently labeled affinity reagents, allow them to incubate. We then rinse off the unbound affinity reagents and then take a picture. This picture, this digital starry sky, is really the primary data that comes off of the platform, and it recognizes that a given probe bound at a given coordinate. We then wipe off those affinity reagents and come in a second step with a different reagent. Again, bind, take a picture. And so cycle after cycle, we're touching each protein over and over and over again with different reagents, giving us additional pieces of information about each individual molecule. This is done in a massively parallel fashion, again, billions of molecules at a time. How do we use this information to actually identify proteins? This is really where the platform branches into having 2 different modes of operation. In general, there is one core platform, and it can operate in either a discovery mode or a targeted mode. The difference is simply what types of affinity reagents we use to probe the sample. For the discovery mode, we use a novel class of affinity reagents, which we call multi-affinity probes, as shown here on the left. For targeted applications, we use the same platform, but switch to site-specific and target-specific probes, often that are commercially available. In fact, by simply reusing the millions of available antibodies, we can extend the functionality of the platform to studying proteoforms of important molecular targets. Before I describe how we utilize these off-the-shelf reagents validated for proteoform analysis, let me first describe how the platform is used in a broad-scale manner. Now, what might be intuitive for how we might identify a molecule, we can think of it as playing a giant game of Guess Who. We have a molecule immobilized on the surface, and we want to know which molecule it is. One way that we could ask that question is by using a series of very specific reagents and asking exactly one question. Are you a chameleon? Are you a cow? Are you a lion? But as you can see, this is incredibly inefficient. And to ask an individual question for every single protein in the proteome would take A, a very large number of questions, and B, a very large number of reagents. But there's another way to potentially identify proteins, particularly if you're allowed to interrogate each molecule multiple times. And that really is what's so special about the platform, is that for each molecule, we're able to ask many questions of it. So instead of using highly specific reagents, we can instead ask much more general questions like, are you a mammal? Are you a large mammal? Are you a cat? A woodland creature? And so in a very small number of questions, one can get to a very specific answer about what molecule is present at a location. Our equivalent of this is using multi-affinity reagents that are not protein specific. Instead, we use affinity reagents that target very short epitopes within proteins, typically 3 to 4 amino acids long. The workflow is very similar to what I showed you before. We mobilize our proteins. We then come in and interrogate with these very special multi-affinity probes that each recognize, again, a short epitope, typically 3 to 4 amino acids long. Take a picture. We then wipe off those probes and then come in with another affinity reagent that recognizes a different epitope. Now, while each individual probe is not protein specific, the combination of multiple affinity reagents, the combination of multiple touches on each molecule is sufficient information to infer the identity of the protein. And so after the data has been collected, we can then ask the question, all right, for this individual molecule, we had binding from probes that recognize this epitope and this epitope and this epitope and this epitope. And then ask the question, given this observed pattern of binding, what protein is compatible with that observed pattern? This is a pretty sophisticated machine learning problem, but intuitively, you can see that some of the proteins might have many, many, many touches, and using that information, you can disambiguate it from other proteins. One question we often get is, how does one build and characterize affinity reagents of this sort? This is a very unusual class of affinity reagents. And this has been the result of a tremendous amount of effort over a long period of time. And in fact, inside the company, we have a very, very extensive affinity reagent development team. For many of the reagents, we first developed them using a process called phage display, where we where we select for affinity reagents that recognize short epitopes. That leads to candidates, which we then test to verify that they in fact bound to the peptide target that they were selected for. That's the first layer of characterization, is just to make sure that they in fact bind to what they're expected to. Next, we examine how these potential affinity reagents bind to a variety of other epitopes and to proteins containing those epitopes. So for instance, shown in the upper center here is the binding of this particular probe, which recognizes the epitope WNK, to the protein PARP1, which contains that epitope. We next test each of these probes against a library of millions or billions of different peptides that contain a variety of epitopes to ask the question, what is the range of epitopes recognized by a given probe? In addition, I should mention that in addition to collecting all of this information, we're collecting kinetics information, on target, off target, and building up a detailed map that says, all right, well, the probe was selected to recognize the epitope WNK, but it also has some ability to recognize, for instance, phenylalanine NK. That is perfectly great that these affinity reagents recognize a wide range of biosimilar epitopes. But we do a tremendous amount of work to understand that and characterize that. This is in contrast to many other affinity reagents that you hear about, where really they haven't been extensively characterized nearly as much. We put a tremendous amount of effort into this. We also can look and verify, all right, in proteins, are these able to bind, potentially even in the context of what once was a secondary structure region or a buried region in a protein. Again, we do denature the proteins during our assay. I'm sure you'll have more questions about probes, and I look forward to answering them in our Q&A. Now, I'll just quickly show you an example of using this PRISM method, protein identification with short epitope mapping, to experimentally identify a set of proteins. In this experiment, we started with a handful of proteins, including transferrin and glucose 6-phosphate, and performed an experiment where we looked at these proteins across a set of 36 distinct multi-affinity probes with a different target epitope per cycle. What you're seeing here in the bottom is the potential, the theoretical binding probability for each probe against each of these proteins. And what you can see is that there are some probes that are actually quite specific to an individual protein. So for instance, the probe in cycle 17 is quite specific to just transferrin. On the other hand, there are other probes, such as the one in cycle 14, that bind equally well to G6PI and transferrin. So while that particular probe is not a great differentiator amongst those two proteins, it does help differentiate those two proteins relative to this third model protein. This really is the substrate for the machine learning algorithm that a priori has a sense of what probes might bind to which proteins. As I mentioned, this experiment was collected over about 36 cycles, taking over 20,000 images. We validated that the proteins stayed immobilized across the experiment. And what you're looking at on the right are a very small snapshot of these digital starry sky images. Again, the primary image data. Even this very small experiment was about 100 gigabytes of data. After collecting this raw image data, it's next boiled down to a binding matrix. In this binding matrix, for each individual spot, so each row of this matrix corresponds to one individual molecule. And what you can see as you go across is whether we called a binding event in a given cycle to a given probe or not. You can see that there are many different patterns that are present. Ultimately, for any given protein, there may be a wide variety of binding patterns, not a single one. And that's because this is a highly stochastic single molecule system. Just to show you the 5 most prevalent binding patterns for transferrin and G6PI, that's what you're looking at here. And you can see that there is some variety, though there are some particular probes that bound to every single instance. There are others where it bound some smaller percentage of the time. You can also see that there are some probes that are quite specific to transferrin versus G6PI, and then there are others that are undifferentiating. Again, it's really the totality of being able to probe each molecule over and over and over and over again that allows us to infer the identity of each protein. If we go inside the machine learning, what we see is something like this, where we're looking cycle after cycle at, in this case, one molecule of transferrin, and asking, all right, what are you? And what you can see is that there's information both in binding and lack of binding. And as we go cycle after cycle probing, shown are where their binding events are these triangles, you can see that we get an increase in our confidence that this protein is transferrin cycle after cycle after cycle. Likewise, for another protein where we had a different binding pattern, we see an increase in confidence that it's G6PI. One question that we often get is how many cycles, how many affinity reagents are required to substantively decode the entire proteome? Simulation results suggest that instead of needing tens of thousands of affinity reagents, that this will be possible using on the order of 200 to 300 affinity reagents. And it really comes about because of the combinatorial nature of each probe being able to recognize many different proteins. Once proteins have been identified, the next step is quantification. And as I mentioned up front, this happens simply by counting. We can start with identifying each molecule on the array and assigning a confidence to each identification, and then essentially counting up each of those identifications to get a quantity of how much of each protein is present. A simple demonstration of this can be done with 2 samples that have our proteins, transferrin and PKM2, in a background, and then asking, are we able to recapitulate the quantitative differences between them? And we can ask those questions in both a relative quantification question or in an absolute quantification question. In a relative quantification question, we might look at transferrin and note that between sample 1 and sample 2, there's a 6-fold difference between the abundances, and we get that back. Likewise, we can look at PKM2 and see about a 2-fold difference between those. What's also exciting is that one can potentially also look at transferrin versus PKM2 in the same sample and recognize that there's a 2-fold difference there as well. One of the very exciting potentials of a single molecule platform is its sensitivity. To begin to investigate that, we performed a very simple titration, looking at transferrin in a background and asking what the effective limit of detection of of our assay was. What you'll note in this titration curve, which is just the amount of transferrin immobilized in the chip versus the measured signal, is that we're able to quantify down into the yoctomole range. I promise you this is not a term that I made up. This is a real term and really speaks to the incredible sensitivity of the platform. Just to sum up the broad-scale platform, it's really been exciting sharing these new results with you, highlighting the development of this robust of the robustly and highly characterized multi-affinity probes, showing examples of data from increasingly large numbers of samples, speaking to the stability of the platform, demonstrating the sensitivity of the platform into the yoctomole range. Now, I'm going to change gears and talk about the other aspect of the platform, the targeted aspect of the platform. And this platform, again, relies on the same core architecture, however, a different set of affinity reagents that are now off-the-shelf reagents that are validated for targeting either individual proteins or individual sites or modifications within those proteins. And what I'm so excited about is that we talked about at the beginning how advances in biology and our understanding of biology and disease have arisen when technologies have pushed the boundaries on resolution and scale. And what's so exciting about these initial, very early findings is that they're pointing to expanding our ability to do proteome analysis, to look at proteins with a resolution and scale that simply was incredibly challenging to impossible previously. In particular, being able to look at a combination of multiple splice variants and post-translational modifications on a per molecule basis. Just for context, what we're going to talk about are termed proteoforms, and they're critical drivers of phenotype. As you're familiar, when DNA is transcribed, it often is transcribed into a variety of splice variants, which give rise to multiple protein isoforms from the same gene, often including or excluding particular cassettes. Proteins are then functionalized, modified with a variety of post-translational modifications, methylation, acetylation, phosphorylation, and more. A proteoform is really the product of those two variants. So one gene may give rise to hundreds or thousands of different proteoforms. Again, these variants of each individual gene-derived protein, these chemically distinct variants. And it's believed that these variants are critical drivers of the function of the protein within the cell. Now, when we look at existing techniques, oftentimes those techniques are looking solely at the presence or absence of post-translational modifications. They aren't looking at full proteoforms, the exception to this being top-down mass spectrometry. So for example, this is a very common workflow used in shotgun mass spectrometry, where we take our proteins, digest them, enrich for particular post-translational modifications, and then do a shotgun proteomics analysis. And this allows us to look at individual sites and say, all right, well, this is how much of that site is modified. But unfortunately, these methods lose context to be able to say, oh, well, how many molecules were doubly phosphorylated or triply phosphorylated? Additionally, it can also be challenging to quantify how much was the protein abundance overall changed versus the abundance of each of these variants. This is most easily understood in looking at this diagram, where we have two samples represented. The first sample on the left contains one molecule that's triply modified, and then two molecules that are unmodified. The sample on the right contains one modification at a different site on each of the molecules. Biologically and functionally, these are likely very different from each other. The molecular heterogeneity of the proteoform landscape is just different. Unfortunately, any peptide-centric method will be unable to tell that these two samples are different from each other. One needs to look at an undigested protein to really look at proteoforms. our first study diving into this is looking at a protein called tau. And oftentimes when one looks at variation in tau, and particularly isoforms, our common tool is the Western. But the Western hides a lot of detail. Under the hood of this Western are a number of variants of different splice variants and post-translational modifications. Unfortunately, these two species, for instance, that are both doubly phosphorylated but in different locations, they have the same mass. All of these here likely run at approximately the same location on the gel and are indistinguishable. And so, again, we're losing detail, we're losing resolution, and really unable to dig into the biology. What we really want is we want to be able to extract from our sample perturbed biologically and say, all right, well, this is for this particular form that is the 0N4R isoform and is doubly phosphorylated, about how much of that specific proteoform is present in our sample. And how does that change in response to perturbation or disease progression? Now, I want to pause there for a moment, just because I'm really excited to share with you in the next couple slides something that is really hot off the presses and really has not been widely seen before. And this really is the first time that we're diving into variation in tau and the biology that potentially comes from that. Now, these are very early pilot experiments, but they are, I believe, illustrative of the kinds of questions that we're going to be able to ask in the future. As I mentioned, we're going to dive in focusing on the protein tau. We began with tau as a partnership with Genentech. It's a critically important protein in neurodegenerative diseases. It's also a great example for studying proteoforms because it contains already a wide 6 different isoforms defined by inserts in the N-terminal region and the C-terminal region, the 0N, 1N, 2N, and the 3R and 4R variants. But then on top of it, work such as done in the Steen lab has elaborated a wide number of post-translational modifications on top of that. Unfortunately, the stoichiometries of these modifications, the combinations of these modifications, the patterns really has not been elaborated to date. In the Nautilus platform, the way that we do this is by using highly specific reagents. So to identify each molecule and determine its proteoform, the way that the platform works is, again, very similar. It's the same core platform. We perform our sample library preparation method, we immobilize the molecules. We might then introduce a pan-tau reagent just to figure out where all the taus are on the surface. We then can introduce site-specific reagents to light up where all of the phosphothreonine 181 modifications are. And then wipe it off, come in again, and look at the phosphoserine 214. And by doing this over and over and over again, one is able to gain increasing detail about each molecule. As a first test of this approach and this platform, we began with a simple control mixture. In this mixture, we started with a recombinant protein that was the 0N isoform of tau and a recombinant 2N isoform of tau. We then phosphorylated the 2N with an enzyme protein kinase A that fairly specifically modifies just serine 214. We then took the 0N form and modified it with an enzyme ERK2 that is both overly promiscuous and also incomplete, modifying about 90% of 181 and 396. We then used a panel of about 5 affinity reagents, allowing the exploration of 32 different proteoforms of tau. So we took our control standards, we mixed them together, and then we were able to investigate what the composition of the mixture was. Now, this is a bit of a crazy plot, so I'll just explain it a little bit. Each row of this corresponds to one of our affinity reagent channels that we're looking at. wherever you see a black circle, that corresponds to where we had, that represents the form that contains that modification. So for instance, if one were to look at this particular species here, this species would correspond to the quantity of protein in our sample that was the 0N isoform, doubly modified at 181 and 396. So to validate that the platform was behaving itself and was able to make a accurate measurement, you can see the amount of material that was the 0N form. You can see the 2N form. One can then look and see this individually phosphorylated at 214 with our very precise pKa. And then one can also see the amount of that 0N doubly phosphorylated. I'll also note that these sites are very far away from each other, so they would never be caught on the same peptide. But as I mentioned before, this ERK enzyme is both incomplete and promiscuous. And so it really wasn't clear what the other 10% of molecules were. But you can see here, you can see those incomplete phosphorylations, and you can also see that promiscuous phosphorylation. This was a panel looking at 5 reagents and measuring about 32 proteoforms. Ultimately, what we've expanded the panel to be so far is 11 different reagents, which enables the measurement of 2,048 proteoform groups of tau. And these span both a range of different isoform reagents, as well as a series of multiple different phosphorylation sites. Now, this really is the first data that we're presenting with precise proteoform level information. And what we're going to show you first is a little bit of validation of that, and then measuring that in biologically relevant model systems. This new data highlights our platform's proteoform level analysis of tau, and this is something that simply wasn't possible at this scale and with this sensitivity previously. First, we did want to verify that one was able to have quantitative measurements. For this, we mixed different proteoforms recombinantly together in different ratios and validated that the quantification was accurate and reproducible. Next, recognizing that tau is an incredibly low abundance protein, we wanted to be able to demonstrate that an enrichment assay, a pull down, could enrich the amount of tau in a sample without biasing substantially the proteoform composition. This assay uses a panel of multiple affinity reagents, pan-tau affinity reagents, targeting different sites within pan-tau. And what you can see here on the x-axis is we had a recombinant mixture of multiple proteoforms, mixed them together and measured them. We then put that sample through our enrichment process with this multi-antibody pull down and showed that there was no significant alteration in the estimated quantities, suggesting to us that we were not substantially biasing the proteoform composition through using the enrichment. And this is important, again, in real biological samples where tau is an incredibly low abundance protein. In Alzheimer's disease, there are a number of critical questions being asked. Things about causal relationships and what drives the progression of the disease, questions about aggregation, questions about model systems, and on and on. And we're really excited to begin exploring how proteoforms contribute to these questions. In our first study, we wanted to look at how the progression of disease is impacted by exogenous perturbation. One of the big questions is about the protein A beta and its interaction to drive hyperphosphorylation of tau, which ultimately drives disease progression, cell death, etc. To do this, we performed a study using iPSC-derived organoids. So these are progenitor cells transformed into neurons, grown as organoids. And we treated these organoids with amyloid beta at multiple time points and multiple doses. These treatments are known to induce tau-dependent pathology and amyloidosis. We used a panel of probing reagents to look at the proteoform landscape in response to this perturbation. What's amazing about these studies is that we're able to look at proteoform level information in a biologically relevant model system for the first time in response to a biologically relevant perturbation. And we're able to unpack multiple levels of detail. The first question, and I want to emphasize that this really is a first look at the data. Before we are able to make definitive biological statements about these kinds of studies, these need to be done with a substantially larger diversity of samples, a larger N, and increased technical biological replicates, et cetera. However, I'm just so excited to share these with you because this really is the first look at this kind of data and illustrates the kinds of questions that might be able to be asked on the platform. So the first question one might ask is just about the isoform landscape. And what one can see is that isn't substantially altered. One can then get to that next level of detail and start looking at individual phosphorylation modification sites and see how those are impacted by this A-beta treatment. Now, one sees a slight increase in phosphorylation, not at every site, but some slight increases. Now, where this gets really exciting is looking at how there are multiple modifications per molecule. This is a measurement that simply cannot be done in other ways. And one can see a potential shift to the right in terms of an increase in the prevalence of multiply modified species, particularly double and triply modified tau molecules. Further diving into the level of detail, one can look and say, all right, well, how does this change over time? Let's look at variations in particular proteoforms. And this is, again, a little bit of a complicated plot. Each row corresponds to one specific isoform. The 0N other is almost certainly the 0N3R. We just didn't happen to include a 3R antibody in this experiment. And then each column corresponds to a pattern of multiple modification. So again, the proteoform in this upper left-hand corner would be the 0N4R isoform that has been quadruply modified at each of these sites. And broadly, one can start to see composition texture variation in the proteoform landscape as induced by the treatment of neural stem cells, neural organoids with amyloid beta. Really exciting to be able to look at that diversity, that multiplicity of proteoforms, and not all proteoforms are present as expected, and not all proteoforms change substantially. But that's where it's exciting to be able to dig into this biology. One other study that we performed was really getting to the question of model systems. This is a huge challenge in Alzheimer's disease. Human brain collection can often lead to technical challenges and variation, and it's unclear where you might want to use an iPSC-derived organoid versus a mouse versus one needing to use human tissue or something in the middle. And we were very fortunate but many of the questions that are asked of these model systems tend to be grossly phenotypic, looking at things like fibril formation or cell death. Can we instead maybe go a little upstream and understand how this model systems differ from each other in their proteoform landscape? To do this study, we looked at 3 specific model systems. We looked at, again, iPSC-derived organoids. We also looked at a model system from a group at Mount Sinai that is called MyBrains. These are a multicellular model system. So unlike the organoids, these organoids that only have neuronal cells, these model systems have up to 10 different types of cells, so a more realistic model system. And then we also looked at humanized mouse brains. Again, we can start digging into detail and start saying, all right, well, how does the isoform landscape itself change? And you can already see some differences between the different model systems at the isoform level. Now, of course, this might be detectable by Western, but this is the lowest level of detail. One next can look at the individual phosphorylation sites, and absolutely there appear to be variation between the mouse brain and the organoids, with the organoids being potentially more phosphorylated, which makes sense. They're further in their Alzheimer's disease progression. Likewise, when one goes that next level of detail to be able to look at multiple phosphorylations per molecule, again, you can see this shift as one moves to the more disease model systems, the organoid and the membrane, where you start to see this multiple modifications per molecule and an increased prevalence of it, potentially associated with progression. One can also then dig even deeper and start to look at each individual proteoform and examine and say, oh, well, it seems like there's potentially more of this proteoform present in the iPSC organoids and the membranes. Again, I want to be really clear that in order to really dissect this biology, a larger number of experiments are required. But this is a tantalizing first look that suggests that there's potentially really exciting proteoform biology to be found. So I'm going to just wrap up now that we have developed an assay for the quantification of potentially thousands of tau proteoforms using the platform. These millions of single molecule measurements suggest that there is exciting biological variation associated with tau pathology and across different model systems and in temporal response. This was enabled by single molecule quantification using tau epitope-specific affinity reagents. And what we're really excited about in the future is both validating and extending these findings with larger study sizes that are appropriately matched to effect sizes, but also going further with even more different model systems, perturbations, time points, etc., as well as potentially looking at additional sites and maybe ultimately different proteins as well. As we've discussed today, the Nautilus platform has two very exciting modes of operation: a broad scale mode aimed to measure the width and depth of the proteome, and then a targeted mode that is intended to provide an incredibly high resolution view into specific targets of interest in specific biology. We anticipate that this will unlock tremendous insight across a number of applications, ranging from mechanism of action studies, such as we discussed with tau, as well as opening up opportunities in biomarker discovery, validation, target discovery, and more. With that, thank you very much for your attention today. I'm really excited to dive into questions now.
Thank you for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located in the lower left corner of your screen. We'll answer as many of your questions as we have time for. Let's go ahead and get started. Our first question asks, can the targeted proteoform mode accommodate targets other than tau?
Thanks so much for the question, and thanks everyone for joining us. The answer is yes. The platform is incredibly general. And so we've done a lot of work specifically on tau, but we've also looked at other proteins, including targets like P53 and EGFR. And so really the constraint on the proteoform, the targeted proteoform analysis, is really just about what affinity reagents are available. And so proteins that have a number of different post-translational modifications or splice variants and have affinity reagents targeting those sites, we've been able to incorporate many off-the-shelf reagents. Really what we do is we do a lot of work to characterize those reagents to ensure their specificity, their binding kinetics, their non-specific binding, and how they react to being fluorescently labeled. And so long as all of those things are good, we are able to incorporate them in the platform.
Excellent, thank you. All right, our next question that we have here asks, are we able to detect all amino acids using the probes?
Thanks so much for the question. I'll just refresh that our probes recognize very short epitopes, typically 3 to 4 amino acids long. So they don't recognize individual amino acids. Within those those 3 or 4 amino acid segments, we have been able to develop probes that recognize every amino acid within there. We haven't built all 8,000 possible trimer probes, but we have built a wide variety of probes across a wide range of amino acids sequences. The one we are a little careful, I'll just note, with probes that contain lysines, because our sample preparation method preferentially modifies lysines. We're also a little careful with probes that contain cysteine, just because they are so frequently modified. And so while we have been able to generate a wide variety of probes, we are particularly careful against short epitope probes containing those 2 amino acids.
Excellent, thank you. All right, our next question here, we have time for just a couple more. So our next one asks, how do you remove high affinity antibodies after each cycle?
Fortunately, our proteins are glued down to the surface and effectively through the nanoparticle. And so we can be incredibly stringent with the buffers that we use for removing bound antibodies, including incredibly high affinity antibodies like the tau antibodies. So we've used a wide range of buffers to remove. We've used buffers that are, for instance, 10% SDS or high concentrations of guanidinium hydrochloride. Because the proteins are immobilized, we have a lot of flexibility there, and that allows us to pick incredibly stringent buffers. Now, one place that we are often very careful is that we try to avoid using buffers that are potentially damaging to the, or removal solutions that are damaging to the proteins. So for instance, we try to avoid things that are either very, very strong bases, which might damage the surface, or very, very strong acids, which might damage the proteins. But we found that a wide range of removal solutions are highly effective.
Okay, great, thank you. All right, and we have time for maybe just one more question, so I'll go ahead and ask this one. This asks, how is the technique altered if the probes recognize more than 3 to 4 amino acids?
So it really isn't. When any given probe is fluorescently labeled and binds, the fundamental data from the instrument is those digital starry sky images that I showed earlier. And so really what the heart of the platform is being able to immobilize individual protein molecules, probe them with any affinity reagent that is fluorescently labeled, take a picture, and then iterate. And so if those probes recognize whole regions of a protein or 3 to 4 amino acids or post-translational modified regions, the actual assay is identical. Now, the reason that we chose 3 to 4 amino acids for our broad scale offering is that turns out to be the ideal length that balances the efficiency of decoding a substantial portion of the proteome with the specificity required and the tolerance for lack of specificity. So for instance, if one built affinity reagents that recognized 5 or 6 amino acids, you would need a lot more of them to decode the whole proteome. On the other hand, if one built affinity reagents that targeted 2 amino acids, they would have to be perfect. They wouldn't be if they recognized off targets or biophysically similar amino acids, their power to decode the proteome would go down. We discussed this extensively in our white paper that's on bioRxiv about the method. So for more detail about why we chose 3 to 4 amino acids, I encourage you to take a look there.
Wonderful. Thank you, Parag, for your time today and your important work. We would also like to thank LabRoots and our sponsor, Nautilus Biotechnology, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period will be addressed by our speaker via the contact information we provided at the time of registration. So please continue submitting your questions as they will be answered. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Goodbye.